Welcome to Vaccine Design course. Here we'll be dealing with the step-by-step processes in one shot. Okay, I'll just explain. So the first step that we'll be doing is the retrieval of antigenic protein sequences. This comes under primary protein structure prediction of antigenic protein. I've written down the step-by-step procedure. Please have a look at it. So we'll be going to the NCBI protein database to retrieve the antigenic protein sequence. The next step is to check the antigenicity of the protein. That is, to predict the functional properties of the antigenic protein. First step is to check the antigenicity, and then is to check the allergic nature of the antigenic protein. Okay, so after retrieving the protein sequence and checking the antigenicity and allergenicity, the fourth step is to study the physiochemical properties of the antigenic protein, as it will help in the wet lab experiments. Okay. Now, after studying the physiochemical properties of the antigenic protein, the fifth step is to predict the secondary structure. Now, after predicting all the functional parameters, we'll be going to predict the secondary protein structure of the antigenic protein. Okay. After that, after predicting a stable secondary structure, we'll be predicting the functional analysis of the antigenic protein structure, which provides a functional prediction as well as the domain prediction of the particular protein. Okay. After the functional prediction as well as the domain prediction, our next step would be considering a protein which is antigenic, non-allergic, which has stability in terms of physiochemical parameters, and which has more number of domains. Our next step will be to predict the epitopes. So we'll be considering a protein which has a lot number of epitopes, because epitope or antigenic determinant is a really important factor in this particular reverse vaccinology design. Okay, so here we'll be dealing with several other softwares to determine various epitopes, like the linear B cell epitopes, the conformational B cell epitopes, the antibody specific B cell epitopes, discontinuous epitopes, and various epitopes like the, to predict the immunogenic regions, and also to predict the glycogen antigen epitopes. Then we'll be looking to predict the cytotoxic T cell epitopes, also various T types epitopes like the MHC class 1 as well as MHC class 2 epitopes. So for that, we'll be looking for different servers. Then, so that comes under the functional analysis of antigenic protein structure. After predicting the epitopes, we'll be selecting the protein which has more number of epitopes, and our next step, the eighth step, is to perform the homology modeling, which comes under tertiary protein structure prediction. So after tertiary protein structure prediction using automated as well as alignment modeling, you can use either of it. Okay, alignment or automated modeling. What we'll be doing is the functional analysis of this particular antigenic protein model that we got out of the homology modeling. For that, the ninth step would be to check the stability of this antigenic model. And then, since we have found out the antigenic model, we need to find the antibody model. So we'll be doing the antibody modeling, and then finding out the stability of this particular antibody protein model. And the 12th step is to perform the antigen antibody docking or molecular docking. That's the 12th step that we are going to perform, where we'll be looking for the two servers to do the antigen antibody docking and then visualizing it in a server. Okay, so these are the 12 steps that we'll be doing in vaccinology or vaccine design course. Thank you.